So now let's move into labeling, okay, with isotope tracers. And this is a really handy approach. Actually, we use mixing models for a lot of different things. And this is the basis of actually what a lot of what goes on in geochemistry. And so are you guys all familiar with this? Can we just like brush it off, or should we take a minute to solve the equation? No? Yes? Okay, well, I'm going to sort of force you to do it. Okay, so let's just let's just do it really quick. Okay, so we have this mixture. The total amount of C13 times the concentration is equal to some combination of new stuff and old stuff, right? And there's too many unknowns here, right? So the trick is we set this total concentration to 1, and we just assume that there's these two parts, right? And then we solve for the F, where the fraction new is equal to 1 minus the fraction old, right? And so in our examples, it might be from a label, it might be from a C3, C4 vegetation shift, okay? And so, okay, then when you're trying to solve for the amount of change in the pool or the flux, you just multiply the fraction old or the new by the total mass. So you just separate your terms, right? It's 8th grade algebra. I know you guys remember that. Oops, going backwards. I know I'm going to mess it up.
I think you missed it should be one
Am I, oh, this is,
I think the one to the left, it should be, isn't it FNDN?
FNDN, yep. There we go. That's the mistake, thank you. Right. Okay. Thanks for your help. Did everyone get that? Can you see that over there? I'm going to just, well, actually, I can probably raise this. Can I raise it? There we go. And so I redo this every time, right? Whenever I'm working out my next spreadsheet, I just redo it. I just double check, because I always screw it up. And a sign change, or, you can screw up your calculations even if it's simple, maybe especially if it's simple. Good. So and so this is kind of like the bread and butter of mixing. A two-part mixing model is sort of the bread and butter of isotope geochemistry, and it's really useful. But there's some warnings, right? Do you guys remember what the assumptions are of mixing models like this? Only 2 end members, right? So we think of the old and the new as being the end members, and we assume there's only 2. What else? No other.
I was saying no other inputs or outputs, but that would also be.
Right, and so no like reactions, right? No reactions, no other sources.
The end members have to be different.
And the end members have to be, and that's sort of like a requirement maybe rather than assumptions, but there has to be enough separation between the end members. If they're too close together, it's not going to work. Good, and so we need to be a bit careful about our about applying mixing models, because especially kind of back to soil organic matter, really soil organic matter isn't just these 3 discrete little pools. And we can't necessarily say it's either old or new because it's a continuum. It's a continuum of different residence times, right? And so what does that do to our mixing model assumptions? It breaks them. Okay, so we need to kind of, I think, recognize that we are working in this system using these tools where we're not necessarily holding up or verifying the assumptions that we're making. Okay, so we need to recognize, in other words, all mixing models are simplifications no matter what your system is. Okay, so we can, was there a question?
Yeah, are you actually measuring the new and the old, or are you making assumptions about those and then measuring the result of that?
Okay, yeah, good, thanks. Let's just go back to this for a second, because this is important. So we think of the total, so we think of the total, like the mixture, as our main piece of interest, our main, like a main study organism or a compound. And if we're measuring this, we sometimes make some assumptions about what the end members are, the new, the delta new, and the delta old in this example. Okay, the thing is that we, of course, in most instances, we have to measure these. In some instances in paleo studies, we can't measure them. We have to make some assumptions or, kind of, educated guesses about what these end member values might be. But, whether you're measuring them or making assumptions about them, they're always going to be associated with some variability, right? And so when I talk about the error propagation, you need to really, and if you're really going to solve your mixing model properly, you need to propagate the variability in your end members and in your mixture. Okay, and there's a lot of pretty tedious Taylor series expansions that's involved in all that actually. Yeah, so if you want to do it correctly, you do need to account for that. Did that answer your question? Good, so when so let's say we've measured our soil organic matter, we've got the fraction, let's say we've done the fraction old and we're trying to understand its mean residence time. All we need to do is just use a simple exponential decay model and just plug in the carbon amount, the mass of new carbon or old carbon, either one it would actually work, at time t and the initial carbon content, and then we can basically solve for the mean residence time. And we can do this on different kinds of soil carbon pools or any kind of pool in your ecosystem, really. And so this is a way that we can use isotope analysis to understand a little bit more about the stabilization processes that are going on in soil organic matter. So what's happening with the y-axis? It's increasing because, so look, is that an absolute scale? It's a relative scale, okay? It's increasing because with time, because, you guys can figure this out, it's increased by 10 per mil, right? Okay, that's a lot, okay, and that's a clue.
It's greater recycling of carbon.
Sort of. Right, so before there was a C3, maybe forest or something growing there, and then I think this was from France, and then they planted C4 crop into it. Right, and so so we've kind of imposed sort of a tracer, right? This is not just microbial recycling. We've imposed a tracer in the form of a C3 to C4 cultivation shift. Exactly. And so that is leading to this big increase in delta C13, not just 3 or 4 per mil, right? And so what else can we say about the processes here?
The smaller particles have a faster turnover.
Do they? Okay, good. So let's look at that other axis there. So they calculated the turnover rate and the change in the turnover rate. So there's a lot of calculations that go into, there's a lot of raw data that are underlying this figure, right? So the turnover rate is, where is it faster? In the larger particles, in the particulate organic matter, right? And it's slower in the fine fraction. Okay, so does that mesh with what we've learned so far? Totally, yeah, totally.
Can you explain the turnover, I
Okay, so that is just another way of thinking of the mean residence time. So mean residence time is the inverse of the turnover rate. Okay, and so the turnover rate is inverse time. Mean residence time is time in the numerator. Okay, good. And so obviously this is important for predicting soil carbon sequestration because we can understand a little bit more about the fine fraction being the location of the older, more stable or more protected soil carbon. And the particulate phase fraction is turning over faster. Okay, good. And so this is another view on that same study. But it's also incorporating, showing you some more of the data, the raw data, that has, I think, some important points going back to the n-member thing and the mixture and the mixing model, error propagation. So this is the coarse fraction. This is the fine fraction. And so under maize, we have the standard deviation is about 1 per mil, but under C3, when there was no change in the cultivation, it's a quarter of the variability, right? So when we have this change in the cultivation pattern or imposing a tracer, it also often imposes variability, which has an effect when you're doing your air propagation and solving your mixing model. In the fine fraction, actually, even though you can see that it looks sort of like there's more variability than here, but look at the scale, right? It's actually quite a lot less. And this amount of variability is similar in the C3 and the C4 components, right? And so this is sort of a visual illustration that should help you to recognize the importance of error propagation, and also hopefully reinforces the point that if we have sufficient n-member separation isotopically, we can detect actually very, very small inputs or changes in our pool sizes. Okay, good. So now I want to switch gears a little bit and talk about rhizodeposition and rhizosphere processes. What's the rhizosphere? The area around the root, the rooting zone, the soil plant microbe interface, the biogeochemical hotspot. Really amazing location of a lot of really cool research that's getting going. So it's very hard to study because it's so microscopic and so dynamic. But there's a lot of different things going on there with roots putting carbon into the soil and feeding their associated microbiomes. So this was a little study I did with elevated CO2 using the tracer that we got from the tank gas, which was depleted in C13. And we were able to see over 5 years of this study in a grassland in Colorado. Well, the first year we had rhizodeposition that was really high in both the ambient as AC and elevated CO2 as EC. In both of those treatments because of the perturbation of just starting this experiment. I'm going to tell you a little bit more about it. But then in the next few years, the rhizodeposition in the elevated CO2 was about doubled compared to ambient CO2. So that is suggesting that climate change with increasing atmospheric CO2 can increase the inputs of carbon into soil from rhizodeposition, from carbon leaking out from roots, from root turnover, from microbes that are associated with roots, all of those things.
What was EC again?
Elevated CO2 is EC. That was the elevated CO2 treatment, and it was sort of like this experimental setup, and I'll show you a couple pictures in a little bit. And the ambient CO2 had about half the rate of carbon inputs into the rooting zone. Here's some new work that a friend of mine has been really spearheading. The stable isotope probing. So this study here was done because of a tracer C13 that's studied back in that I published over a decade ago. There's some newer work that's being using a similar principle, labeling plants and finding that C13 or O18 or N15 in the DNA of the microbes in the rooting zone. Okay, so this is really cool work. It's really hard. I haven't done it really. I've worked with people who are doing it, and it's sort of new and expensive. So don't get your hopes up really a lot at this point. But the idea here is there's these labeling chambers where they're feeding C13 into these grasses. They also can use N15 enriched litter and O18 enriched water to label, and here's just a close-up of the rooting zone from those plants, and you can sort of see this whole little whitish fuzz around the roots. That's part of the rhizosphere. And so this paper, I think I put it on your reading list. Here's another example. So this is just sort of like a kind of a cool conceptual, gee whiz stuff to show you. I'm not going to show you really a whole lot of data or anything. And this is not using, so what they do is centrifugation, right? So this is not based on any kind of isotope ratio mass spectrometry. They basically label the plants. The plants leak that C13 out into the soil rhizosphere. Then they take the soil, and there's the red line there is the bulk soil. The blue line is the rhizosphere soil. And so the bulk soil hasn't gotten labeled. They pretty much can see that. And then they do this fast centrifugation with like cesium chloride or something. So they get these density separates, right? If anyone else has done DNA work, that's the kind of the foundation of extracting DNA. And so they can see these higher density fractions are the fractions that are taking up the C13 from the plants. And so then they can go and just take that fraction and sequence it, right? They can do primer-based sequencing with 16S, or they can just do the whole metagenome. It's all somewhat expensive, but in this way they can identify microbes or even genes that are using these root exudates. Here's another piece of that same paper, which is sort of a review paper, where they actually labeled the microbes with O18 by adding water, just doing in little tiny incubations. And basically they can get the growth rate because they can see there's this big density difference in the unlabeled and the labeled treatments. And so they can take that raw data and turn that into, just looking even at the whole total DNA, they can look at the new growth rate. And so here we've got time, so they sample it at different points in time, and they can get the growth rate of microbes. And by looking at the total and subtracting out the of DNA and subtracting out the surviving microbes, they can get the mortality rate. And so getting the growth rate and mortality rate of microbes in like a living soil, this is something that is a bit of the holy grail. It's really, really hard to study this in any other way. And then, of course, in addition to looking at the total DNA, you can sequence it and go on. And some of the work that they're doing is looking at microbial food webs, looking at symbiotic relationships like inside termite guts, all sorts of crazy things that you can do with this. Succession of microbial community species. Yeah?
What kind of fractionation might there be in DNA replication?
So this particular example is not quantitative. That's a great question. The question was related to fractionation of the DNA. There's another paper that I haven't studied as carefully where they're doing quantitative QSIP, and they're accounting for the fractionation processes and doing a more of a mass balance type approach to try to account for that. Right? And so I can point you to that reference.
What about sources of carbon in the medium and stuff?
Sources of carbon in the medium, in the growth medium? And that's why you have to have, or oxygen or nitrogen, that's why you always have to have a control. Right, you always have to have a control, and you're running like 2 sets. Whenever you do any kind of isotope labeling or tracer work, you always have to have an unlabeled control. Did you have a question?
So they quantify the culture growth by qPCR? How they quantify the growth in time?
The growth rate is just by, extracting the total DNA, probably qPCR, right? Of the different of this heavy fraction at different points in time. Okay, good. So this is just sort of a really little snapshot. It's not my area of expertise, but this is sort of like, you know, I have sort of research jealousy of this whole field. It's really, really cool. It's growing, but it takes a lot of money and a lot of dedication to get not a lot of numbers out. Okay. So, what else is new, right, with isotope work? Okay, so isotopes provide insights into soil organic matter pools, turnover rates, and carbon stability in soils. Isotope tracers can be useful to assess carbon dynamics, so the process rates. And also we're starting to look into this new era of stable isotope probing of DNA to be able to really understand a lot more of what's going on into rhizosphere processes. Of course, this could be applied to other ecosystems, estuaries, other biogeochemical hotspots where we can learn a lot about how carbon is being cycled through ecosystems in response to climate change.